Howdy and welcome back to Protein Function, Myoglobin and Hemoglobin. We are going to be focusing on hemoglobin in this video, and we're going to be bringing back that word allosteric, other site. Other steric site, okay, or structure. So the other site, not the oxygen binding site, affects the release of oxygen, and that's what we are talking about when we're talking about allosterism and hemoglobin. Protons promote the dissociation of oxygen and the conversion of hemoglobin from the R state, the oxyhemoglobin, back to the T state, the deoxyhemoglobin. And the effect of protons is called the Bohr effect. So we're going to be talking about, in this video, the relationship between carbon dioxide, which is produced in your tissues through the citric acid cycle. So metabolically active parts of your body, such as your tissues, produce carbon dioxide, and as a side product, protons. And the protons and carbon dioxide help to convert oxyhemoglobin to deoxyhemoglobin, and that binding of protons to oxyhemoglobin cause a conformational change that makes it harder for the hemoglobin to bind to oxygen. Because remember, the T state, deoxyhemoglobin, does not easily bind to oxygen unless you're at very, very high concentrations of oxygen in the lung artery interface, right? Okay, so one of the allosteric effectors of hemoglobin is protons that are built up as a consequence of metabolically active tissues. All right, so we're going to talk about the details of the Bohr effect and how it actually works. As the pH decreases, in metabolically active tissues, the pH is going to decrease. The dissociation from hemoglobin is enhanced. And if we ignore the actual stoichiometry of the reaction that's happening, you go from the oxyhemoglobin, that's the R state, plus protons, to deoxyhemoglobin, the T state, bound to the protons, and therefore no longer having a high enough affinity for oxygen in the tissues. Remember, the tissues have a lower concentration of oxygen, and so hemoglobin is already starting to lose its affinity for oxygen, but it's helped along by the Bohr effect. Okay, the Bohr effect, as you'll learn when you listen to Hank Green explain it in the crash course video, helps release oxygen to metabolically active tissues. All right. Christian Bohr, the father of Niels Bohr, the atomic physicist that you've probably heard of, and both Nobel laureates, Smarty Pants family, discovered the effect of protons, that is, the lowering of pH due to higher proton concentration, on the release of oxygen from hemoglobin, and that's why it's called the Bohr effect. Binding of protons, H+, diminishes the oxygen binding on hemoglobin. Even though the protons are not trying to bind to the heme, they don't bind the heme. That wouldn't make sense. That would be two positively charged ions, and they would be repulsive to each other. It binds to a site outside of the heme. It binds somewhere else on the protein. The proton binds somewhere else on the protein, and that's what makes it an allosteric effector. Conversely, binding of oxygen diminishes proton binding, and I'll show you a cycle that explains that. And this has important physiological significance for the oxygen transport protein, hemoglobin, but not for myoglobin, which is not affected at all by protons. Okay, so here's what's going on in the Bohr effect. You'll need to learn this slide. At the tissue capillary interface, that's where the partial pressure of oxygen is lower, you have a hydrated carbon dioxide, which is being produced from glycolysis, and it's the citric acid cycle that's giving you all of these carbon dioxides. And the hydration of carbon dioxide produces an extra proton, which has an allosteric effect, increasing the dissociation of oxygen right where it's needed most, in metabolically active tissues. At the lung artery interface, now coming back up from the T state and needing to bind to oxygen again in the lung artery interface, the reverse reaction is happening. Bicarbonate dehydration, which is required for you to turn carbon dioxide into a gas so that we can exhale it, consumes an extra proton, promoting CO2 release from the heme iron and oxygen binding. So carbon dioxide and oxygen hitch a ride on hemoglobin on the same site, but the protons that are around or that are depleted can regulate the ability of one to bind preferentially over the other. All right, so here's what's happening. Here's carbon dioxide, which is produced at the tissue capillary interface thanks to oxidative metabolism. Here's the water that's around that's dissolving the carbon dioxide. We're not carbonated. That would be a bad thing. Okay, so carbon dioxide is soluble in water, and thanks to carbonic anhydrase, gives you carbonic acid, which is soluble in water, and bicarbonate ion dissociates a proton. Okay, so this is a weak acid, and so part of it is dissociated like this, and some part of it is protonated like this. Okay, so the production of carbon dioxide, therefore, increases the proton concentration, resulting in a drop in pH that you have at metabolically active tissue, and that's binding of these protons to hemoglobin far from the heme site, changes the conformation to deoxyhemoglobin, which can then bind to carbon dioxide and carry it back to exhale it at the lungs. And this is what's happening in respiration. All right, so here we're talking about, and you can spend some time looking at this figure because it actually explains a lot in a small amount of space. Hemoglobin and myoglobin in oxygen and carbon dioxide transport. That's what's going on this slide. It's called respiration. Oxygen is reduced to water in the electron transport chain. So the oxygen that you inhale is not part of the carbon dioxide that you exhale. It is part of the water that you make in the mitochondria when you are making ATP. And the carbon dioxide that you exhale is produced from the breakdown products of metabolism, fat and sugar specifically. And they need to be exhaled, those carbon dioxide molecules. Okay, so here we are inhaling down here at the bottom. Here's the oxygen. It's going to bind to hemoglobin at the lung artery interface. Here the orange is the lung. And we have a high concentration of oxygen in the lung at 100 torr. And that allows oxygen to bind to one monomer of the tetramer hemoglobin, shifting it to oxyhemoglobin. So oxygen binds deoxyhemoglobin, makes it oxyhemoglobin, and then it goes on to the tissues. Once in the tissues, in this case the muscle, we're looking at the muscle tissue over here in blue, the oxygen is let go thanks to the metabolically active muscle tissue, the Bohr effect, the protons, which is happening up here, we'll get to that in a minute, convert oxyhemoglobin to deoxyhemoglobin. The oxygen gets picked up by myoglobin, and myoglobin is going to hold on to that until stress, right? Fight or flight, and then it will let go in the muscle. Now, in respiration, when you have metabolically active tissues, you create carbon dioxide, which is shown here in blue. Remember, the oxygen you inhaled was red, so this is different. Carbon dioxide is produced from metabolism. It is converted to bicarbonate ion and proton. And the proton can protonate the hemoglobin and stabilize deoxyhemoglobin. So this hemoglobin is going back and forth between deoxyhemoglobin and oxyhemoglobin. And the deoxyhemoglobin is stabilized in the blood, in the capillaries, in the tissue. That's what helps. That's a Bohr effect. That's what helps to release oxygen from the hemoglobin in the tissues. And then as you approach the lung, that proton is required to release the carbon dioxide and produce a water molecule so that you can exhale that carbon dioxide product of metabolism in the lungs. Okay, so enjoy learning how to interpret this cycle, this transport cycle. Hank Green is happy to help you. He calls it the vein train in that video on Crash Course.